So this is Cell Division Lab 3 by Darius DeVitt and Bobby St. Pierre. So the introduction to meiosis. The purpose of mitosis is to grow and basically increase the number of cells, because for every one cell you have, you then make two daughter cells. And the majority of the cell's life is spent in interphase, which includes G1S and the G2 phase. The other phases of the cell cycle are mitosis and cytokinesis. In G1, that's where the cell grows, it gets basically ready to divide. In the S phase, or the DNA replication, in the name, the DNA gets replicated, and so that it can stay diploid in the cell, so that the cell can stay diploid. And then the G2 phase is basically just to make sure that everything's ready, because after the G2 phase, there's no turning back. That's the point where cell division is going to happen. It's just a matter of time. And so there are different phases in mitosis and later on cytokinesis, which I will go into later. And so meiosis, on the other hand, is all about sexual reproduction. That's when there are two gametes, one from the mother, one from the father. That's the egg and the sperm right there. One has 23 chromosomes and the other has 23 to make the diploid number 46, as everyone knows. And so there's two steps to meiosis. The first, it goes from one diploid cell into two haploid cells where the homologous chromosomes are separated. And in the second step, that's where it becomes 4 different daughter cells, and then the sister chromatids separate. So it goes from haploid to an even simpler haploid, which I'll get into later. And so in interphase, this is what I explained before. This is the G1S and G2 phase. So basically, the DNA hasn't been formed into sister chromatids yet, and that's why it's all jumbled up. There's still the nuclear envelope, and everything is just normal. And so this is prophase, where now you can see the chromosomes have formed, the nuclear envelope is starting to break apart, and this is where mitosis really starts. Well, this is where mitosis does start. In metaphase, you can see that the chromosomes line up on the equator vertically right here, where in meiosis you will see a difference. And it does this so that the sister chromatids can separate. So in anaphase, as you can see here, what the one chromosome was is now 2 chromatids that are split apart, and that happened for every single chromosome. And in telophase, you can see they've all reached to one side of the cell. So this side has one set, this side has the other. Each are identical at this point. And in cytokinesis, you can see the cleavage furrow coming, and this is where the 2 cells split apart and have 2 completely identical cells. Now, this is where where meiosis comes in. And I have set this up so that you can see each possible way for each step to go at the same time. So let me explain. On the right, you can see crossing over taking place. Right there, hopefully my mouse shows, and right there. And you will be able to watch that play out. And that's called the crossing over. And hopefully you will be able to watch that play out throughout the entire video. And on the left, you will be able to see, as things go on, the different ways that the different ways that things can pair up, and how maternal does not necessarily always have to side with paternal, and vice versa. So after prophase 1 comes metaphase 1, and as you can see, the chromosomes line up a little different. So as you can see in the top one, these two, because they're homologous, they line up next to each other so that they can be split apart. And so what's different about this from mitosis is that instead of being all vertical, now some are side by side. And so as you can see on the bottom left and bottom right, these are just this is just showing the different ways that they can order themselves. So you can see here, there's one paternal and one maternal and on one side, and here they line up the same way. So here's a picture of anaphase 1, well, three pictures actually, where you can see the crossing over on the top and how that is playing out so far, and you can also see on the bottom the different selections showing. So now this is prophase 2, where you've seen in anaphase 2, in anaphase 1, they separated, they got into their own little sectors, and that pretty much split apart, where now in prophase 2, they're two completely different cells. So you can see in the top, they have their own little crossing over effects, still differing from the other two. And so just to clear things up, the blue pen was dying, and because we did not have our pictures that were done during lab, I decided to take a whiteboard and I drew all of this out. And of course, with my luck, the blue pen decided to die. So as you can see in the top right here, I decided to make a key where the green is maternal and the once was blue, now is red, is paternal. So just to clarify all that. So as you can see, in the bottom there is this is just following what I've been saying before, how maternal and paternal can go together, or paternal and paternal. I mean, it doesn't really matter, because as long as the homologous chromosomes aren't together, then everything is fine. Because as you can see, there's this big chromosome and this little chromosome, and the way they match up has nothing to do with each other. So here in metaphase 2, just like in regular metaphase and mitosis, they line up top to bottom, head to tail. So you no longer see the side-by-side pairing anymore. And so now they are lined up at the kinetochores, at the centromeres, and as you will see in Anaphase 2, they are now beginning to split apart. They are coming together, and like they're coming farther and farther apart, where you will see now they are 4 completely different daughter cells. So let me just go back. In this cell, it was divided into 2. This cell divided into 2 to get 4 completely different cells. So in the top, you can see the crossing over effects made a significant change in both the second 2 chromosomes. I mean, in both the second and third cells. And in the first and third, they are just regular daughter cells. And just another way to order everything and the different possibilities. You can see if you follow the left side, you can see how the tall red is matched with the short green, and vice versa. Or if you look on the right side, you can see all green are matched with all red. And after this, you can continue to watch our case study. Thank you.